Hello, my name is Emily Song, and I am a sophomore at the College of Arts and Sciences studying neuroscience. Today, I will be giving a brief summary of my poster, which explored the effect of lesioning the insular cortex on taste and feeding behaviors. I am currently working in the Bentley lab under the mentorship of Professor Nicholas Bentley, and our lab focuses on hunger and exercise and the neural circuitry involved with and in hunger and food-seeking behaviors, which have greater implications for eating and exercise-related disorders in humans. I chose to specifically target the insular cortex as one of these possible circuitries, as the insular cortex is implicated in the integration of sensory, emotional, and cognitive aspects of feeding behavior, supported by a strong connectivity to extensive brain regions. This study explores the insular cortex's role in modulating adaptive feeding behaviors and taste aversions through a series of behavioral experiments on C57B16 mice, comparing insular cortex loss of function and control groups. Initially, after lesioning the insular cortex via a ibotenic acid injection, adaptive feeding tests with high-fat and high-sugar diets indicated a tendency for a reduction in high-sugar diet intake in insular cortex lesion mice, suggesting altered post-ingestive feedback. Expanding on this, subsequent two-bottle tests with sucralose, a non-caloric sweetener, versus water, followed by sucrose, a caloric sweetener, show no significant preference differences in lesion mice compared to controls. However, in tests comparing sucralose and sucrose, the lesion mice displayed a tendency for reduced preference for sucrose, implying disrupted post-ingestive sugar feedback. Another behavioral paradigm, the conditioned taste aversion test, further highlighted the insular cortex's involvement in taste and visceral pairing, with lesion mice exhibiting attenuated aversion to aversive stimuli, specifically in the sodium chloride intake tests. Through multiple behavioral experiments, we found that insular cortex lesions led to a tendency for decreased preference for calorically dense foods, for example sucrose, compared to non-caloric sweeteners, for example sucralose, indicating a disrupted integration of gustatory and metabolic signals involved in adaptive feeding decisions. This is suggestive that the insular cortex modulates post-ingestive feedback mechanisms and taste aversion, contributing to altered dietary preferences and intake behavior. In terms of future directions, the brains of the insular cortex lesion mice will be collected and Nissl staining will be conducted to visualize the lesion in the insular cortex, allowing for the mapping of the location and magnitude of the lesion, ensuring the intended insular cortex cell bodies lost function. In following experiments, the sample size will be increased to enhance statistical power, and we will utilize different experimental approaches, such as neuron activation through photophotometry, in order to explore the functional and anatomical connectivity of the insular cortex and how it influences taste and reinforced behavior. To conclude, I would like to thank both CURF and the PURM programs, who provided the institutional funding for this project through the CURF grant for faculty mentoring undergraduate research. Thank you.